This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 24, recorded February 23rd, 2011. Hi, everybody. I'm Vincent Racaniello, and with me is Dickson Despommier.
Hello, Vince.
Welcome, Dickson.
Thank you.
Good to see you again.
Good to be seen.
Dickson has a royal blue shirt.
This is true.
With ruffled cuffs, which makes him look like royalty.
I don't have ruffled cuffs. No. This is a
What
standard. No, these are little pleats or something.
Pleated, sorry, not ruffled. I don't know
ruffled.
my fashion.
It's pleats.
It has a texture.
It does.
Interesting.
It's herringbone. It's a herringbone style.
How have you been? I hear that you have been around the world. No, you've been to
Not
London.
quite. Just to London.
I see London. I see France. So what did you do in London?
Well, interesting you should ask, Vince, because I was there to give an after-dinner talk to a group called Global Food Safety Organization, the Global Food Safety Organization. And it was sponsored by an American company called Diversey, operating out of Racine, Wisconsin. But this meeting is now 10 years old, and in the first 2 or 3 meetings, they told me they had something like 30 or 40 people show up for it. And now they're in their 10th year, and they had to turn people down.
And what's the subject of this
Food
meeting?
safety, Vince.
Did you tell them not to use human fertilizer?
I did, but in the meantime, I told them the story about the very first foodborne infection discovered.
Was that Jon Snow?
No.
That was
Keep
something.
guessing.
The first foodborne?
Now, how would I know that, since I only did work on Triconella spiralis?
Was that the first foodborne infection?
Triconella spiralis, yes, it was actually.
What year was that?
It was exactly, I know the exact year, it was 1835.
First foodborne infection.
Identified.
No. So 1835, though, is after Louis Pasteur, so we know about bacteria by then.
No? They didn't. They did not.
Louis Pasteur was when?
It was a little later than that.
So, and Robert Koch was also later.
That's right. So the germ theory of disease had not yet developed.
So end of the 1800s.
That's right.
So they just said, in this case, there's something in the food that's making people sick, but they didn't know what it was, do you, Mr. Jones?
That's right. So Mr. Richard Owen, and I've already told this story on an earlier episode of TWiP, how Triconella became discovered.
Right.
And it was discovered in England, right next door to where this meeting was being held, and nobody at the meeting knew the story, so they were fascinated by the story.
I must say that I am also guilty of ignoring history.
We all are.
I try not to. But
yes, we all are.
Talking to a group interested in safety of food, as a parasitologist, you could talk for hours and hours and hours.
You might say I had the meeting out of my hand, but I would rather not have the meeting out of my hand. The meal, however, you can imagine what a meal would be like at a food safety meeting.
Was it safe?
It was awesome, too. It was awesome and safe, and it was quite wondrous.
Wouldn't it be funny if there were a foodborne outbreak of illness?
It wouldn't be the first time, Vince.
At such a meeting?
It wouldn't be the first time.
At least it wasn't on a cruise ship.
This is all true.
Because if it were, they would have a good chance of getting viral gastroenteritis.
That's right. Cruise ships are famous for Norwalk virus.
Norwalk and rotavirus
and
is also. Calices, well, that's
and
Norwalk.
There was an outbreak of Triconella on board ship once, too.
Really?
On a cruise ship. Yeah, sure.
Well, a cruise ship is a good place because it may not actually occur more frequently on a cruise ship, but they are monitored.
Yeah, I know.
By public health authorities.
That's right.
So we pay a lot of attention to them.
This is true.
But, new people come on every week or so.
Yep.
And there's it's a closed environment. It's a good scenario for passing on infections.
You bet. So let me ask you this, Vince. You've traveled a lot.
I've never been out of New Jersey.
He lies. His nose just got longer, everybody.
Oh, yeah, I lie because I'm here in New York City right
now.
not
only that, you were in Paris recently, and then you went to Seattle. And how did you go there, Vince? You probably didn't walk.
I was a virus. I see the world.
He teleported.
I traveled in a host.
No. How did you get there?
I've been
Vince?
around. I've been in various
you
places.
have.
I have not been in every country.
No, I mean neither. Not even close to it. I
I've
haven't
only-
even been to every European country.
Yeah, I haven't either. I've never been to Africa.
But to get there, you have to fly, correct?
I flew.
So, do you know anything about the pilot and the co-pilot?
Do I know anything about them?
Yeah.
That's a really vague question,
Do you know what their
Dickson.
duties are?
To fly the airplane.
Well, yeah. Well, the pilot flies the airplane, right? And so the co-pilot sort of sits there.
They eat peanuts.
No, they don't eat peanuts, Vince.
They make sure the food is safe. They make sure they don't bring infections around
When
the
the
world.
stewardess serves the meals to
I think
the
they're called
pilot,
flight attendants now. You're dating yourself.
so it's fine. Okay, fine. I'll go back and cancel the date.
Flight attendants.
When the male or female flight attendant brings the meals to the captain and co-captain,
Right.
co-pilot.
Oh, they eat it first before anyone else?
I keep guessing.
They eat it after everyone else.
No, there's something basically different about their meals. They get different meals.
So that if one is sick and the other is not.
This is true because food poisoning can occur on an airplane, too.
They should get it after everyone in the cabin has, but then it would have to be rapidly developing. It wouldn't work. So what do you mean separate meals? They're
different.
They're all nuked.
They're totally different. Yeah, but that doesn't kill off the enterotoxins from E.
Right.
coli.
But on the ground,
Or
they're prepared in
salmonella,
different
or-
kitchens, you mean?
They try to ensure the fact that both the pilot and co-pilot eat different food. So
Okay.
in case one of them does get gastroenteritis that's so acute that he can't fly the airplane, the other person can.
Yeah. That would have
Or
to be
she.
from a toxin because
Yeah, no, that's right.
the replication time
course.
would needed for
No, toxins.
something else.
It's all about enterotoxins.
Dixon, we
Yes,
could do a podcast on food safety, but this is
We could do that.
TWiP.
Oh, but you know what? TWiP has a lot of food safety
Yeah, I
issues
know
in
we
it.
talk about. I just don't want to keep talking.
No,
about
I
this.
understand
This
that.
is
Maybe
really-
we'll do one in the future on that.
But what do you? You're going to teach a course in urban agriculture,
I'm
right?
going to try to do that. Yeah. I'm
Maybe
gonna
you
try
could
to
do a
that.
podcast as part of the course. You should do a podcast on urban agriculture.
Perhaps.
I have a farm on my roof. End of podcast.
That's right.
All right. Well, this is TWiP, This Week in Parasitism.
Yep.
I could actually just edit it to this point.
You could do that.
Right,
That's
but
right.
People,
They want to
may
hear
or-
more.
Maybe they want to hear what you've been up to.
They want to hear more.
That you've been talking about food safety. And you talked about it from a parasitological perspective, I presume.
Well, actually, I talked to him about from the
Bacteria
perspective
as well.
of the vertical farm project.
And that takes care of toxins also in your foods as well.
That's true. That's true.
All right, today on TWiP. Last time we talked about the little dinosaur with all the armor on it, Ankylosaurus.
We did. No, Vince.
You remember?
I do.
What was the name of that?
That was Encelostoma caninum and Encelostoma brasiliensis and
No,
Encelostoma
you're in the wrong...
duodenale.
Didn't we talk about strongyloides?
We did.
So ankylostoma is hookworm.
That's right. That was before that.
Encyclostoma, no?
No.
You say encelostoma?
Encelostoma.
Oh, okay. Here, 2 episodes downstream, I learned how to pronounce it properly. Nicator americanus.
Not a problem.
Hookworms. Then we did Strongyloides stercoralis, right?
Correct.
Which is horrid.
It was a bizarre worm in
It's
terms
just
of
a
its
worm that
life
is
cycle.
a big problem in people getting immunosuppressed, right, for transplants.
That's right. It's still going on today, too.
All right, so these are all nematodes.
Yep.
A lot of nematodes. And now
lot
today,
of nematodes.
another nematode is ready for us.
Another nematode.
What would that be?
Well, we're going to switch into the filarial parasites now.
Filaria. Why are they called filaria?
Because of their shape.
The other ones look like a worm.
Yeah, I know that.
They're
but
round worms.
there are two different kinds of shapes that they apply to the configuration of nematodes. And they're complicated terms derived from Latin. Okay, one is called rhabdidiform. Okay, and the other is called filariform. So you have two different shapes of worms. The filariform shapes are narrow and thin, and the rhabdidiform shapes are stumpier and fatter. And Caenorhabditis elegans actually has a stage of his life cycle that gives you those two different shapes of worms as well.
All right.
So, in fact, the rhabdidiform larva of Strongyloides is not the infectious stage of the infection. The filariform larva, which is the third stage of the worm, is the infectious stage, and it's based on its shape. Okay, so filaria are narrow, thin, thread-like worms.
I have to tell you, Dickson, I'm looking at a picture and it looks the same as every other worm we've looked at.
But you know what? Viruses look like that to me too, Vince. But
No.
They're all different. Yeah, but from some level, they all do look like little small particles.
Well, they are small.
That's true. But worms up close have remarkably different shapes and patterns. And the best way to look at a nematode is through a scanning electron microscope.
And in your book, you don't have any such photographs.
We do, but not of the nematodes. We have some of the trematodes, and they're remarkable pictures because you get to see every little crevice and pore and mouth part, and that's not true. We do have some of nematodes also. If you go back to the hookworm chapter, you can see the cutting plates and the teeth on Ancylostoma. Those are scanning electron micrographs done by David Scharf, by the way, who's a genius at putting these pictures of parasites and other objects too, like mosquitoes, etc., into the eye of the public so that they can actually see what these beasts.
These are colorized, these pictures.
They're colorized, that's right.
To make them dramatic.
But you can see all the detail this way.
Sure.
Okay?
No, scanning EMs are great.
At this point, you wouldn't think that these are similar at all. You'd say they're all different. But from another size perspective, they do look the same. So it just depends on how close you get to something before you start to see the differences.
All right,
so today we're going to talk about a group of organisms that differ in life cycle from all of the other parasites that we've discussed so far, except the vector-borne protozoans. Can you imagine, Vince, a worm that you can see with your naked eye?
Being vector-borne?
Yeah.
Must be a big vector, Dickson.
Well, actually, it's a pretty small stage of the worm that's transmitted by vectors.
Right.
And there are
that makes
several
sense.
kinds of vectors in this case.
One question.
Please.
Let's just briefly review some of the vector-borne protozoans that we talked about.
I'll tell you what, Vince, let's take a little quiz. Are you willing?
Sure.
I'll give you the name of an organism, and you tell me whether it's vector-borne or not, all right?
Sure.
Now, I'm sure the audience is going to get a big kick out of this one. Okay, Giardia lamblia.
Not vector-borne.
Entamoeba histolytica.
Not vector-borne.
Cryptosporidium.
Not vector-borne.
Leishmania donovani.
Vector-borne.
Plasmodium falciparum.
Vector-borne.
Good, Vince. You're doing well now. Trypanosoma brucei.
Vector-borne.
Trypanosoma cruzi.
Vector-borne.
There you go. So those are the ones.
I listened.
You did. You got 100.
Thank you.
You can now
Dickson,
teach this course.
Part of my goal in doing these radio shows, if you call them that, is not only to teach everyone, but to learn.
Here. No,
and we just started TWiP, and am I going to learn on that one.
Me too. I'll listen. Trust me.
You should come on the show
sometime.
I will,
of course I will.
When we talk about Wolbachia
you know
or something like that.
Either one of us knows probably a thimble's full of microbial life in the form of a bacterial infection.
Of course.
So we could probably supply enough ignorance to the subject to make
questions.
The audience.
But remember, the principles are similar.
Absolutely.
So parasites are not part of microbiology, right?
Of course
are they? So we have a department of microbiology. Would there be parasitologists in such a department?
If that's not a traditional place for them, but
where would they be?
They're in the Department of Parasitology.
What if there isn't? It would be tropical
medicine. That's another place that they stick those people because they don't know what to do with them.
Well, some parasites are.
Tell me about it.
Unicellular.
You have a parasitologist in your department, and I'm in the department also in
microbiology. David Fidduck.
What does he work on?
Malaria.
Aha.
And he's in the microbiology department.
He certainly is.
Question answered. Okay. Wait a minute. In your book, Dickson, it says lymphatic filariae.
Yes.
No, that's not what we're doing today.
Nope. We're going to start off with the one that lives in the subcutaneous tissues.
Onchocerca volvulus?
That's correct.
But that's a filarial worm, right?
Of course it is.
All right.
And the thing that makes it a filarial worm, among other things, of course, they all have general characteristics in common, and one of them is that they're all vector-borne.
All of the filarial worms are vector-borne?
That's correct.
All right.
So, in this case, the first one that we're going to discuss, Onchocerca volvulus, is the primary cause. It used to be the primary cause of blindness throughout the world.
Would that be river blindness?
It would, Vince. And why do you think they call it river blindness rather than mountain blindness or desert blindness?
I would venture a guess that you acquire the infection near rivers. or in rivers?
Vince, you're spot on.
It's not
You are
hard.
spot
It
on.
is not difficult.
In fact, the vector that transmits this is an aquatic insect.
An aquatic insect.
That's right.
I didn't know.
just
What's
like
the name of the insect?
Mosquitoes are aquatic insects, Vince.
So when you say aquatic, it means they spend a good time of their life.
They lay their eggs in water.
Mm-hmm.
The eggs hatch in water. The larvae grow up in water. And the only thing that isn't in water is the adult.
And I would call that an aquatic life cycle.
I would too. So therefore...
and fish are aquatic.
That's true.
And they're always in the water.
They're always in the water, Vince. And alligators are sometimes in the water and sometimes not, but they're aquatic organisms.
How about toads, Dickson?
Toads are mostly land-based, but they have to lay their eggs in water. But you know what? Some frogs don't lay their eggs in water.
Mm-hmm.
They don't. They're remarkable adaptations to the lack of water. That's the reason why they're so unusual.
The vector for Onchocerca volvulus
is
I don't know, what is it?
Well, you're reading it in my book. It's
a
the black fly. That's right, or Simulium. The name for the black fly genus is Simulium.
And where is it only the black fly and no other vector?
That's correct.
And what is the geographic distribution of the black fly?
Oh, Vince, they're found everywhere.
Are they here in New York?
Darn right.
Really?
Anybody who trout fishes and goes up to the Adirondacks to trout fish,
yeah.
If you go up in early June,
yeah,
and you go trout fishing in, let's say, the Osable River,
yes,
near Lake Placid, you will encounter black flies.
I thought this was something restricted.
Oh, no.
Okay, the black fly is sometimes called a buffalo gnat. So
That's true.
How big are they?
They're about 3 or 4 millimeters in length.
Buffalo gnat, turkey gnat, or white socks.
I would never call them that.
White Sox, they're
White
black,
Sox. Those are baseball players. Come on. It's an insult.
There are over 1,800 known species of black flies.
That's right.
We are inundated with insects, aren't we?
We are. They rule as long as microbes let them.
Most nourish themselves by...
taking blood meals.
Yeah.
That's right.
Do the females need blood to lay eggs?
They both take blood, and the female also uses the blood for laying eggs.
So the male also takes a blood meal, as opposed to... some mosquitoes, right?
All
mosquitoes, the male doesn't take blood meals?
Not at all. In fact, the female doesn't either. She feeds on plant juices.
She just needs it when she lays eggs.
That's correct.
So if you have a mosquito bite, you can say this was done by a female mosquito.
You bet you can. That's right.
So black fly is the vector.
Yep.
And this is a filarial worm.
Yep.
And when was it discovered? In the 1800s?
Yep.
And was it by an Italian of some sort?
I'm not sure. I haven't...
Leuckart.
Leuckart. Leuckart was German. Germany was a hotbed for parasitology research in the 1800s. And in fact, I described an event with regards to Triconella. The life cycle of Triconella, although the worm was discovered in England by Richard Owen, the life cycle was worked out by Rudolf Virchow, operating out of Germany. So the Germans were really involved in lots of different research, and they did a lot of their early work in Egypt. And remember, I described some papers by this guy Loos, L-O-O-S, who Dubini, in the St. Gothard's Tunnel incident with regards to hookworm, discovered his papers and made the connection between the infection and the disease from hookworm, but based on some of these early German workers. And Leuckart was a very famous German parasitologist.
He first described it in Africa.
It's not a difficult thing to describe, though, because it creates a lesion that's palpable. It's about the size of a walnut.
On your exterior surface.
Yeah, it's underneath the skin. So I've got some pictures someplace.
So there's no intestine involved in this life cycle, is there?
None whatsoever. It's either in the blood or...
Here's a nodule with worms in it.
That's right. And you can see we've depicted some of them over here. And if you go online to the Google Images section and just type out Onchocerca, you'll see lots of them.
Okay. So where should we start with Onchocerca?
Well, let's start at the beginning, of course.
Okay.
How do we acquire it?
Okay, then we'll get into how we
the pathology
treat
of
it. The pathology.
And it's got an unusual twist, Vince.
How we prevent
it. It's got a TWiM twist.
TWiM twist? Is there bacteria involved?
Well, yes, there is, Vince.
Good. And
Wolbachia.
Really?
Oh, yeah.
Excellent. Is it good or bad to have Wolbachia?
It's horrible for you. And
good for the
perhaps
worm.
Good for the worm. We're not sure yet what it means for the life cycle of this parasite, but
interesting.
Yeah, it's very interesting. So we'll get to that.
Okay, so beginning is the life cycle, right?
Right. So how do you acquire Onchocerca volvulus.
Well, we've already said the vector is a black fly.
That's
so
right.
if
So
you
tell me exactly how we acquire it now. In your opinion, if this is a biting midge, which it's not, but it's closely related because they're all dipterans, and this thing bites you, you would make the assumption, probably, that the parasite is injected into you during the act of feeding. Is that true?
I would have, but now I'm looking at this and I see it's not true.
That's right, it isn't.
Okay. It isn't true.
But most people make the mistake of thinking that all vectors inject things into you in order to get you to become infected. And in fact, there's another infection group which is transmitted in exactly the same way that the filarial parasites are transmitted. Mm-hmm. But they're not worms, they're protozoans. In fact, trypanosome, cruzi, and a bunch of other related trypanosomes in Central and South America are transmitted not through the bite of a reduvid bug, mm-hmm. but rather through the feces.
Right. We mentioned this.
We did, of course. So it deposits the organisms on the skin, and then somehow you have to rub it into the bite wound that the insect makes.
So the insect, while it bites you, it's defecating, right?
That's correct, to make room for the blood meal that it's taking.
Right. And then it flies away, and then you scratch it or something.
That's right. So in this case, these parasites are in the biting mouth parts of the fly.
It's picked it up from another host, right?
That's another person. That's
person
correct.
Always? Is it always people involved?
Always.
No animals?
There are no reservoir hosts for Onchocerca.
Only people.
Just people.
And there are no species of Onchocerca or related ones that infect animals? There are.
No, there are. Sure, there are.
But they don't go into people.
That's right.
And vice versa.
This is exactly
right. Okay. That's interesting. We've had some crossover before among
these
true. Parasites.
But in this case, there isn't. Thank goodness, because otherwise you would be very hard to eradicate it.
Sure. You think we can eradicate this?
Yeah, I do. This is an eradicatable disease. You can do this. And in fact, nobody knows this better than your wife, Vince.
Yeah.
Why is that?
Well, because the drug that her company developed called Ivermectin is very good at eliminating this.
That's the one.
Onchocerca, and it's been used to combat river blindness.
And it's been used to eliminate the parasite from certain countries. As long as they cooperate with their epidemiologists.
I think Bill Gates should get involved with this. Shouldn't he pay for the drug? Merck was actually giving it to them, right?
They were.
Do they continue to do that?
Yeah, they do. Yeah, they do.
So why don't we give polio vaccine, which is a penny a dose?
I don't understand.
Ivermectin, I'll bet, is more than a penny a dose.
It's a lot more than that, but Merck has made a tremendous amount of money on the animal parasite
sure, it's for
sales.
It's called HeartGuard, right?
Yeah, but it's also for lots of other large animal parasites.
Any dog that you want to eliminate heartworm
That's right.
You give it ivermectin. Heartworm is?
It's another filarial parasite, and if you'd like us to discuss that, we will.
Didn't we?
No, we haven't discussed Dirofilaria emitis.
All right.
Not at all. So the parasite is transmitted through the bite of a black fly.
An infected black fly.
It has to be an infected black fly.
And where in the fly is the worm? What does it look like? What stage?
Right, it's third stage.
Stage.
All of the nematodes that are infectious infect as a third stage.
So it enters the fly as a third stage?
No.
As a what stage? Second? First stage, and goes to the second and third stages.
Right, and it develops actually in between the flight wing muscles in the thorax of the insect.
All right. And the third stage are the transmissible forms.
That's correct. So it crawls out of that zone into the mouth parts as soon as it detects the heat from the body of a host.
I see.
So it doesn't go into the mouth parts until the insect is set to feed.
Very quick thing, in other words, because the fly is landing on you for less than a minute.
And not only that, it doesn't stay there, Vince.
What doesn't stay where?
The parasite doesn't stay inside the black fly while it feeds. It crawls out of the black fly onto the skin next to where the bite wound is.
This parasite is actually putting itself into the host, not the vector.
Correct.
So it crawls out of the mouth while the fly is feeding. What does the fly do? He or she is taking a blood.
Yeah, they have these scissor-like mouth parts that they slice through the tissue with.
Great.
And then they create a pool of blood and they suck it all up.
All right, and in the meantime, these
sounds disgusting.
Worms are coming out of it.
It is disgusting. Have you ever been bitten by a black fly? And you know that when they fly away, there's a little bead of blood that develops.
Have you been bitten by black flies?
Vince, I can't tell you how many times.
You go upstate New York
or Maine.
Right?
If you go to Maine, they'll carry you off. They're so intense. That's why the moose in Maine during the summer months, when the black fly season is at its height,
away.
No, they go into the swamps and they just stick their noses out. because it's so intense. No, I'm not
Okay.
kidding. You have to wear mosquito netting when you fish. It's all kind of messy stuff.
So, listeners, anyone have black fly experience? Tell us.
Oh, I'm
So
sure they do.
Is there any Onchocerca in the U.S.?
Nope.
Why?
That's a good question. I wonder why there aren't.
If we brought some here, would it propagate? Because if you
Another
remember,
tough question.
this fly is in Africa. This worm is in Africa. I
Not
presume.
just Africa.
Other places. But we talked in the hookworm episode about how hookworms came from Africa
That's
to the
right.
U.S. via the slave trade.
That's correct.
You would think that these would have come also.
Well, Vince, if we had started discussions with Wuchereria bancrofti, you would have been exactly correct. We had an endemic center for Wuchereria in Charleston, South Carolina.
But not for Onchocerca. Do we know why?
We probably don't have black flies in the South.
Oh, really?
Black flies are a northern thing.
But in Africa,
Why is
they're
that?
in the hot places.
You know
right?
what? That's exactly right. So you're going to have to tell me about black flies a little bit now.
I don't know anything about black flies.
Would you like to know something about
yeah,
black flies?
you're going to have to tell me. Everything I know, I've learned from you.
Well, you're in trouble now then.
Is that true?
Well, everything I've learned from other people, so I can relate their stories to you. And that's basically what we're doing right now, right?
It's fine.
So black fly larvae live in extremely oxygenated, fast-running waters.
Okay.
So think about the South, Vince.
It's pretty slow.
It's pretty slow.
Because of the hookworm, Dickson.
It sure is. It's as slow as you can get.
Dickson, that's a Texas accent.
No, I'm from New Orleans. I should be able to do an accent, but I can't. The point that I'm raising here is that the ecology...
I'm going to New Orleans in May, by the way.
Are you?
Yeah.
The ecology of...
Sorry.
Great. I might join you. The ecology of black fly biology is related to cold, clear, oxygenated water for the most part. For the most part, I said. Now, if you go into the hills of Georgia and Alabama, not Alabama, but North Carolina and Virginia and West Virginia and Tennessee and Kentucky, you can find those streams.
Mm-hmm.
Okay?
Mm-hmm.
But I have a feeling, Vince, that when slaves were caught in Africa by other Africans, by the way, not by the English or the Americans or whoever was around at that point, but the Africans themselves were catching young male black people and selling them into slavery.
Right.
That if they saw lumps and bumps on them,
Oh, they wouldn't take them?
right,
Ah. And that was all you needed to see an infection.
you can see this infection.
By the way, Dickson,
Yes, Vince.
what is the name of this infection?
Onchocerca. Onco.
Right.
It's almost cancer-like.
Right.
That's probably the derivation
That's the
of
worm,
this.
Onchocerca.
Yeah, that's right.
What's the infection called? What's the disease called?
River blindness.
There's another word.
Which one are you looking for, Vince?
Onchocerciasis.
Oh, onchocerciasis. Okay, fine.
We have to mention it at least once.
That's fine. The disease
Because-
is referred to as onchocerciasis. That's true.
Onchocerciasis.
That's right.
And onco because it looked like tumors.
Right.
But it wasn't.
No. The other thing is, of course, if you live in an endemic center in Africa and you're exposed to this infection as a child and then a young adult and then an older adult, the number of bites that occur where the infection is given, if the bite is close to the head, the worms have a better chance of causing blindness than if they're in the lower extremities. So there were some peoples in Africa that wore clothing and others that wore very little clothing. And the ones that wore very little clothing were better off than the ones that wore clothing, Vince, because the flies only had limited areas to bite people.
Sure.
So they would bite them around the neck and around the face.
Yeah.
And when that happened, of course, they would go blind. Not right away, but they would go blind eventually.
All right. So we've
And
established
it's mostly a
that-
West African disease, by the way. There's no onchocerciasis in
In the
East Africa. The Rift Valley actually separates this out. And but of course, there aren't too
All
many fast-running waters
Yeah.
in East Africa.
But in
Most
West
of it is
there
in
is.
West. Yeah, that's right.
All right, but in the north of the U.S., there are fast-running waters.
Tons of them.
So this
We didn't have slavery in the North.
okay. So in theory, if we introduced Onchocerca in the north, it should.
We might have
propagate,
been very lucky not to introduce
but
this.
it hasn't. All right.
All right.
And because the slaves were.
But we
checked
did
for these
introduce
bumps,
another-
It didn't come into the South.
That's right. We introduced, but filariasis in the other form, the elephantiasis developing form, those people would be excluded from importing also because of their disease.
So we've been bitten by a black fly.
And the
And
larvae
the
are
larvae
deposited on the skin.
And why don't they just go into the bite?
Well, because the fly is there.
All right, so the fly leaves, then what
The
happens?
fly withdraws its mouth parts, and the moment that happens, in goes the larva, and eventually gets into the bloodstream. Or, in some cases, they stay put. And in the case of Onchocerca, it stays put.
It doesn't go into the bloodstream.
No, it doesn't have to. It goes into the subcutaneous tissues and molts and becomes an adult. And this adult begins to grow, and you'll never guess how long these adults can get, Vince.
I would say 2 inches. He's holding his hands about 2.5 feet apart. Really? They can grow that long?
Amazing.
What, are they all coiled up under your skin, and that's why you get a bump?
And look at the cross-section of the lesion that I
That's
show
all one
here.
worm?
No, there are probably 5 or 6 worms in that one.
So if you biopsied a lesion, you could pull out a very long worm.
Well, you can't obviously pull it out because it's stuck in connective
break.
tissue.
Sure.
It would break. The point is you need males and females here too, just like you do for the other nematodes.
Okay, let's get this straight. So you're given a stage 3.
Right.
It molts
Twice.
and then becomes either a male or a female.
Right.
And they mate in the lesion
They do.
and make eggs.
No. Well, you have to make an egg first, but let's take this to the next step.
All right, the female makes eggs. The male has fertilized the female inside,
right.
or the eggs are fertilized outside.
No, inside.
The eggs are laid. They hatch.
No, the eggs are not laid. The egg develops embryologically inside the worm, just like Triconella did, to produce live larvae.
All right.
And these are called microfilariae because they're small.
Mm-hmm.
They're microscopic, as a matter of fact. And these microfilariae
Yes.
migrate away from the original infection site.
So they come out of the female.
Yes.
They migrate away.
In the subcutaneous tissue.
All right.
Only. They never get in the blood.
How far
never
can they
get
go?
in the blood.
Can they go 6 feet?
Well, how tall are you, Vince?
I'm not 6 feet tall.
Well, then they can't go 6 feet in, Vince. But I am.
That was a reference to hookworm.
Right. But in this case, you can reference it to the host.
So they can travel your entire length. So if you have a toe bite, they could get up to your forehead.
And vice versa.
And when do they stop?
They don't.
They just keep moving around?
They do.
And what are they doing as they're moving around? Are they destroying
They're waiting
tissues?
for a black fly to come and suck them up.
And that's how they're picked up again. Ooh, this seems like a low-frequency event. How many of these microfilariae do you have in you? You need to have a lot.
Billions. Look at this section of tissue right here, Vince. You can see lots of them.
So if you initially are bitten by one black fly,
You
get a couple of larvae, stage 3 larvae.
Well, you might have more than a couple, but you know,
few hundred?
the black fly itself has a limit to the number of
Yeah.
worms it can suck up and live with.
So these normally do not harm the fly up to a certain point.
Up to a certain point, but at a certain level, they start to kill off the black fly.
Yeah,
so,
which is not good. You don't want to kill your vector, right?
yes, you would. Why not?
Well, then you can't be vectored.
That's right. So what if the heavily infected people of a community were actually sparing the not infected community by simply giving so many microfilariae to the black flies that all the black flies would die instead of transmitting the infection? There are some situations that they estimated where some villages were protected by the heavily infected people, simply from that for that
Kills
reason.
the fly, huh?
Yeah, it kills the fly.
Now, from the few hundred stage 3 larvae that you get, those could bring billions and billions of microfilariae.
That's correct.
And is this a problem for us?
If the microfilariae stayed in the subcutaneous tissues and didn't get into the connected region of the body that connects to the connective tissues, then the answer would be everything would be fine. But
that happens
that's not
often.
what happens.
All right, what's the timing here? If the fly bites you, how long does it take to get an onco, a lump?
Oh, that's months.
You get one.
Yep.
Not multiple.
No, one per bite.
If you're multiply bitten, you can have many, right?
Many.
And then each of those, after a month, give off microfilariae.
No, it's later than a month. You've got the lesion there, and then maybe 3 months later you're starting to get patency. They call it patency.
Yes.
Where the females are actually giving birth to live larvae now, and that can go on for years.
And you can survive with this.
people do survive with it.
All right.
Sure they do.
Do you,
However,
do you itch? Do you have
not
pain of any
without,
sort?
it's not without symptoms. And there are a variety of symptoms associated with the dermatologic aspects to onchocerciasis. You get this condition called peau orange. Peau orange is a French term for orange skin. And this is a West Coast African disease, and so most of those countries speak French, or at least a lot of them. Côte d'Ivoire, all those places, right? So Senegal, a lot of those countries speak French. And as a result, the French scientists investigating the diseases of the French colonies, and this was one of them, applied terms to their patients that reflected their own language. So you get skin disorders, you get exfoliation, you get dermatitis, you get itching, you get a lot of different things associated with it. But there's a big but.
Yes.
There are some connections between the subcutaneous tissues and your eyes. This is unfortunate.
So they can get into your eye.
And they do.
The microfilaria?
Yeah.
In what fraction of infections?
Well, if you wait long enough,
eventually they'll go in your eye.
A lot of them will get in your eye, but it depends on how many of them get in your eye as to what happens next. Mm-hmm. And if you look at the sections of eyes removed from patients who have either died for some other cause or have had eyes removed because of the extreme damage done to their eyes by this infection, you will find that virtually every part of the eye is invaded by these worms. Now,
including the
cornea, everything.
What's the space inside?
The iris, the vitreous humor.
Is it in there?
Oh, sure.
Is it full of fluid, Dickson?
It is. There are two fluids. There's an atrium and there's an anterior chamber and a posterior chamber. The worms live in the subcutaneous tissues, Vince. They don't come out into the fluid, basically. So, but the point is that the longer you're exposed to the infection, the more the hypersensitive reactions are elicited by the presence of the worm excretions.
Yeah, I see.
So you develop this hypersensitivity that gives you all the symptoms. However, I'm holding something back here, Vince, because I led you to believe that the pathology of this infection was not due to the worm itself, but rather to a symbiont that it's harboring inside its own tissues, namely Wolbachia. That's a very recent finding. Wolbachia apparently either causes the worm to produce products that are hyperreactive to the host immune system, or they themselves are the cause of the hypersensitivities that develop. The curing of patients of their Wolbachia, but not of their infections, reduces or eliminates the pathological effects of this infection. It's remarkable. And that's maybe 5 years old. Or less than that. You could go online and take a look at it now and see how many references you have to it. I'm sure you have a lot of them, because this was a totally unexpected finding. Totally. And so what is Wolbachia doing inside of Onchocerca?
Well, it's a
symbiont. Yeah, but I asked what it's doing. I didn't say why it's there. What the heck is that thing doing inside of a parasitic nematode?
It's getting something.
It's not. It's the only one that we know of. It's
inside of Onchocerca?
They are mutually benefiting each other.
But as you know, Wolbachia is in other insects.
Oh, it's in all over.
It's in mosquitoes, right?
It's
Flies,
everywhere. It's everywhere.
In many other places.
It's everywhere.
Yeah.
It's like Chicken Man. It's everywhere.
Interesting organism.
It's a bacterium. And we should actually raise that as one of the main features of TWiP.
Yeah.
Because Wolbachia is a big deal. All right? And there's tons of big deals out there, right? But for Onchocerca, it's the biggest deal.
So it makes it pathogenic, essentially.
It exacerbates the pathogenesis.
So have experiments been done with and without Wolbachia?
Yeah.
And it makes it worse. Is there an animal you can infect with?
This has no animal model for humans.
How do you do these experiments?
You use their own filarial infections, which also have Wolbachia.
Animals?
Yeah.
Okay. No, that's good.
Yep.
I just wanted to review something here, which
Please.
Has to do with the life cycle. You'll enjoy this. Adults.
I wrote it, so
produce
I won't tell you.
Well, you probably don't remember it.
I probably don't remember.
Adults produce hundreds to thousands of microfilaria during their lifespan, about 700 per day.
Okay.
For 8 to 10 years.
Yeah.
What happens after 8 to 10 years?
Well, it's interesting. Why don't we go back to the lesion that's created by the bite of the black fly first to discuss something extremely interesting.
I know what you're going to say.
You do?
It has to do with your guy, Triconella.
That's correct.
Right.
That is correct.
Because I saw it here. I was going to actually say that. Hey, Dickson.
Yeah.
So tell us
the way this worm survives in situ is that
What does that mean, in situ?
It means that it's inside the host, inside of its ecological niche.
Is this pen in situ?
If you consider this room situ, the answer is yes.
Yeah, it's on a desk. That's in situ. It's where its natural place is, or
That's
where
true.
you're looking at it, right?
It's better if you put it into a pen holder. That would be an in situ for the pen.
All right.
What I'm suggesting here is that these adult worms require nutrition.
In position, it means.
In position. Thank you for looking
It's
that
a very
up.
broad thing. I thought some people might not know that.
Oh, okay.
It just means in position. So if you're observing something in situ, it's in position.
It's where they belong. It's their natural habitat, basically.
So the headphones you're wearing are in situ. That's where they belong.
Only when we're on the air.
Yes. Okay.
So, but I've seen people walking down the street with these too, by the way. But it just doesn't have a microphone. They have a Bluetooth sticking out of one ear.
Yeah, that's right.
So the intrigue of this is that there's a cure for this, by the way. An older drug. There's an older drug.
For the nodules?
Yeah.
So as soon as you see them, yeah, but the microfilaria are already swimming by the time.
The drug of choice for the adults
yes.
is Suramin.
Suramin. That's used to treat HIV infections.
That's
it
right.
was tried. Suramin. Suramin.
That's right. And what is Suramin? It's a very toxic drug, by the way.
You know who was developed by what company, Dickson?
I was gonna say Merck.
No.
Janssen.
It's another German.
Jensen.
No, Bayer.
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 The very first chemical company.
It was developed in 1916. See?
Do you know that they also made the first drug ever?
Bayer aspirin, right?
Sure.
Someone said the other day, I did a TWiP in Seattle, and we had a guy from a company. He said aspirin would never be approved by the
He's
FDA
right.
today.
He's right.
Correct?
Yeah. A lot of GI bleeding, a lot of headaches, a lot of tinnitus.
Apparently it's used for treating prostate cancer.
Right.
That's
So
investigated.
what does it do? What is its mode of
Onchocerciasis
action?
And trypanosomes.
Yep, that's right. What's its mode of action?
It's a very interesting molecule. I'm sorry, I'm not ignoring you, Dickson. I'm just looking at
It's okay.
what it's doing.
Not a problem.
I don't know what the mode of action is.
Okay. It's one of its side effects. One of its side effects is an anti-angiogenic side effect.
Angiogenic meaning
It interferes
developing
with blood vessel,
blood vessel
formation.
formation.
That's right.
Mhm.
That's right. So it actually kills off the adult worm by preventing them from encouraging the development of a circulatory reti,
I know that word.
which surrounds these parasites.
That's from the the Triconella.
Triconella, that's
Ready.
correct. The nurse cell has
Yeah.
a circulatory reti, and so
Yeah.
does the adult stage of Onchocerca.
Very similar.
Very, very similar.
So, Dickson, your angiogenic factor from Triconella, is it similar to the angiogenic factor?
We don't have a clue.
Of
Vince,
Onchocerca?
if there's no money out there for research on the important things, can you imagine what there's not money for? This is one of them.
Yeah.
But it would be intriguing, better than intriguing. It would be instrumental in controlling these infections if we could find out.
Do you think humans are destined to understand everything?
They want to, but I don't think they can afford
Can't
it.
afford it, huh? It's a pity.
Well, I think eventually we'll have the genomes of every organism because they're easy to get and they're cheap. So you'll at least know what their genomes look like, but you won't understand how they work.
So what you're saying is in the future, most experiments will be bioinformatics?
Well, they're certainly moving in that direction.
No
If you
more
look at
wet
the
experiments?
look at the latest issue of Science, which is my pick of the week, by the way, it's all about imaging, visualizing data.
Yeah.
That's all.
Well,
it's about.
we're generating a lot of data now.
They don't know what to do with it.
And we bench scientists don't know how to handle it.
So what you have to do is you have to massage it into a visual, a complex visual that you can understand.
But Dickson, you could look at all the proteins encoded in a genome, and you couldn't tell
No, of
which
course
one
not.
is an angiogenic
Of course
factor.
you can't. Of course you
right?
can't. No, not at all. But there are ways of doing that.
So by inhibiting angiogenesis, you can inhibit the growth of this. So
You kill off the adult.
You kill off the adult, and the nodes go away. Is
Well,
that right?
they degenerate, they become fibrotic, and they don't go away. But they can become calcified.
The worms are gone, though, right?
Worms are there, but they're calcified. They're dead.
No more egg laying.
Larvae. No more larvae laying.
Sorry.
This is
No more
ovoviviparous.
Ovoviparous.
Yeah.
All right.
Okay.
That's what I wanted to point out, that there was this angiogenic.
Sure. Well, they
response.
used to give drugs which were corticosteroid in nature to try to limit the inflammation that the organism causes and therefore limit the amount of blindness. And then there were other drugs called diethylcarbamazine, for instance, and lots of other drugs like it, which were used to kill off the larva production. not the adults, the larva production.
Right.
But along comes ivermectin. Bam.
So ivermectin kills adults and
Well,
microfilariae?
Vince, I don't think it kills off the adults. I think it absolutely limits or eliminates larvae production.
Really?
So, yeah, so that means that you have to give this drug once a year, every year for 15 years to get rid of the infection.
All right, how was ivermectin originally discovered?
Oh, it's a great story.
Was it for this worm or something else?
No, you know how it works at drug companies. They set up these generalist screens for discovering drugs, all right? By the way, as a side note, Pfizer is busy closing its antimicrobials division.
Mm-hmm.
That's tragic.
There are no more infections, Dickson. It's not a problem. It's not a problem.
Well, then, why are you inventing another podcast called TWiP? They're going to take over the world, and they'll be podcasting about us.
To review what we knew.
As an extinct species, they'll talk about humans. I'm not kidding. I mean, it's getting desperate out there, right? So we don't have any microbial agents.
Yes, but this is cyclical, Dickson. It'll come back.
Well, I hope you're right.
Once they see there's money in it, they'll come back.
Or once they can't fight
So
off
ivermectin
these
was identified in the screen for what?
Well, it was when they get a drug. There was an executive at Merck, the story that I was told by Bill Campbell, and I'm sure it's a correct story because he was an honest man. He worked there for many, many years, and he was a dear friend of mine. An executive heard a talk given by a chemist at Merck telling everybody to bring back samples for them to ferment and get products and then distribute throughout the company.
Natural products, right?
Yeah, for various, and they had a tremendous number of different screens. When I say screen, I want to know if there's any antimicrobial activity. Is there any antiparasitic activity? Does it cure psoriasis? Does it interface with zits formation and prevent it? Does it cure diabetes? Does it, and they have screens for these things, right? And so they want to test all these possibilities. So when they take a sample and they place it into a fermentation vat, and they do it both aerobically and anaerobically to make sure that they catch most of the groups of microbes that are in soil, for instance, they'll ship the fluid out afterwards to these screens and see if any of them come back positive. And usually they don't. Or if they do, they turn out to be very toxic stuff. Well, this executive was a golfer, and he went to Japan, of all places. Now, can you imagine what a small place Japan is? It is. It's a very small country with a lot of people, and they don't have room for anything. In fact, 78% of Japan is forested, which is interesting. But this guy went to a golf course in Japan, and the price for membership in this club was over $1 million at that time. Per person, of course. So only the very, very wealthy Japanese could play golf, right? But they love their golf. They actually do. If you've ever seen the movie Lost in Translation with Bill Murray, there is a golf scene in that movie, and it's quite an amusing movie. But nonetheless, I'm diverting from my main point here, and that is that they went on the golf course, and the guy kicked up a divot, and this lecture occurred to him. And he asked the guy he was with, he says, "You mind if I take this piece of dirt back?" And the guy says, "You must be joking. You want that?" And he says, "Yeah." And so he took out a plastic bag and put the divot in the bag. He says, "Here, it's yours." Well, fast forward to today, how many billions of dollars has Merck made selling ivermectin?
So that was isolated from a soil.
It was isolated from a soil sample. It's an actinomycete product.
Yeah.
It looks like dihydroxy chicken wire. It's an incredibly complicated compound.
Streptomyces.
It does not cross the blood-brain barrier of humans, thank goodness, because its mode of action is thought to be on the nervous system. So ivermectin has been used for animal diseases, mostly parasitic animal diseases. Sheep, cattle, all those animals placed into a small area develop large numbers of nematode infections that take away from the commercial production of wool and pork fat and all kinds of other things that you can think of, and it's worth a lot of money to prevent these diseases, right? Or at least to keep them under control. So ivermectin was a boon for
So actually, the series is called Avermectin. So they have a lot of derivatives that they mean, one of which is ivermectin.
That's right. He was from Scotland.
Interesting. So ivermectin was used to treat river blindness in Africa a number of years ago. Merck donated large quantities.
They did, but in the beginning it wasn't used for that purpose. It was used for something else. Maybe it was used for hookworm or strongyloides or something like this, and just inadvertently.
Oh, I see.
They found that it also affected this other parasite.
So this is effective against a number of nematodes.
It's generalist. So you're asking me how they discovered the drug. They actually have dogs at Merck that were infected with heartworm.
Mm-hmm.
And so they ran it through that screen, and they discovered that it limited the number of microfilaria in the blood. And in fact, it reduced to 0 eventually. If you just kept giving the drug, and they didn't know what the drug was at that point, so then they take this broth and they split it apart into all of its components, and they eventually isolate this.
So, what is the global burden of onchocerciasis?
Onchocerciasis.
Do we know?
Well, it used to be very large. In fact, it was the leading cause of blindness in the world.
Really? The leading cause?
It was the leading cause.
All right.
Today, trachoma is the leading cause of blindness.
Which is a rickettsial infection?
No, it's
what is it?
Chlamydia.
Chlamydia, yes.
That's not even a microbe. I mean, we don't know what chlamydia are.
I know what they are. They're bad.
They're bad. Of course they're bad, but where do they fit into the scheme of life?
So they say vector control was useful, but also program of donating ivermectin has prevented 600,000 cases of blindness.
More than
in
that,
11
even.
West African countries.
Yeah, that's an old book.
But you should update this book, Dickson.
We should. We actually should.
It's further been estimated that 18 million children born in this area are now free from the risk of river blindness.
That's
and
right.
25 million hectares of land have been rendered free of the disease.
There is an unfortunate consequence of the overuse of drugs, Vince.
Resistance.
You got it.
So we're getting ivermectin-resistant Onchocerca.
We are. We've used it long enough now so that we've actually selected for resistant strains.
So this is only in Africa?
No.
Where else?
Central and South America as well.
Okay. And is ivermectin used there to treat the disease?
Yes and no. It's not free for those countries. Okay, so they have another program there that they call nodulectomy. And what they do is they go into the villages, a team of surgeons and technicians and nurses, and they inject an anesthetic, I should say, around the nodule, and then they with a scalpel, they actually remove the nodule and pop it right out because it's in the subcutaneous tissues. And they can fill a jar up with this stuff in some communities.
Similar to the jar of Ascaris.
Yeah, that's exactly right.
From
Nodules,
Bangladesh.
and they got rewards for that.
That doesn't cure it, though. They still have microfilaria in them.
The microfilaria will live for a long time afterwards, but for a finite time, about 6 months, and then they die.
And there's no more reseeding because you've taken all the nodules.
Well, no, that's not true, though, Vince. They can go back in the fields and catch it again. So it must tell you something about the immune system and how it reacts against this, too. It's very, very poor. So this worm probably secretes immunosuppressive chemicals as well.
So did you ever see a case of river blindness here at Columbia when you worked in the parasitology department?
We never got those cases because they were too acute. But the Eye Institute knew all about them because they knew it was the leading cause of blindness in the world.
So they might get occasionally a patient there.
Vince,
right?
if you think about this, listen to this. The leading cause of cardiomyopathy in the world, what do you think that is?
I don't know.
It's Chagas disease.
Okay.
It's 12 million cases. Infections, 12 million infections, I should say. That's more than any other heart disease cases, all right? And we have not stopped it. What was the biggest cause of blindness? Another parasitic infection.
Onchocerciasis.
Yeah. And yet these barely appear on the radar screen of most Western medical schools because they are not endemic to the Western world. All right? He's using Wikipedia as a resource, folks. This is horrible, Vince.
Well, some things are correct.
Some, but we don't know which.
It says here, onchocerciasis is also known as river blindness.
That part's right.
Okay. I was just actually looking at the fact that there is a song by the same name by Jay Giles' band called River Blindness, and I didn't know that. I wonder if it has anything to do with river blindness.
I doubt it.
So how do you eradicate this? You said you thought it was eradicable.
Eradication starts... Well, they started a program in West Africa of DDT spraying.
Get rid of the vector. Is that possible to get rid of black flies?
What do you think happened, Vince?
That became resistant to DDT.
How about that? Very fast, as a matter of fact. It happened rapidly. So there are now DDT-resistant strains of black flies. So the next thing they tried was to use something called BT.
Bacillus thuringiensis?
That's a TWiM. That's a TWiM subject. Yeah, no, you can see a lot of touch points here. So they used Bacillus thuringiensis to feed to the larvae in the river because they are the world's greatest filter-feeding organisms of all time. And what happens there is that the organism, the bacterium, contains a protein, which when the bacterium is digested in the gut tract of the insect, it crystallizes under acid pH conditions and forms a giant crystal, which then blocks the gut tract of the insect and it starves to death.
Right.
So guess what the insects have done? Instead of having an acidic gut tract, they now have a basic pH gut tract.
Oh, that I'm surprised that's a fundamental change in physiology.
I know, and yet it has happened.
Well, that's amazing.
So, no matter what trick you have up your sleeve, organisms can outsmart you.
Well,
what's the best way to prevent infection, Vince?
What's the best way to prevent infection?
Yeah, avoidance.
Avoidance is the best
Not
way.
vaccines. In fact, vaccines are good, but they're not great.
Just stay at home.
Drugs, forget drugs. The best cure for tropical disease is to live in temperate zone. Because we don't have any tropical diseases. But as global warming occurs now, I'm not going to go there.
Dickson, you could go to West Africa and be at risk for acquiring river blindness, correct?
Still.
I mean, you just go, you get bitten by a black fly.
Still,
what are you gonna do? Would you take a dose of Ivermectin if you went?
No.
Would you take it when you came back?
No.
Have you ever been to West Africa?
No.
Why wouldn't you take Ivermectin?
Why would you? If you don't have anything, why would you take it?
But if there are a few stage 3s in you from that fly...
No, but you'll know it eventually. You'll get a nodule.
Well, why wait for the nodule? Get them before.
Because using drugs without having an illness associated with them is a dangerous operation.
Okay, I wanted you to say that, Dickson.
You shouldn't give a drug for an infection you're not
All
sure
right,
of.
now I'm going to have you eat your words. Because you said if you were going to get a heart transplant, you would take a dose of Ivermectin.
I certainly would. That's an extreme case, folks.
Well, that's treating without an infection.
No, that's true. That's true. I would take it under those circumstances only. I would break the rule at that point. But I wouldn't break it here.
All right. I don't know why, but I guess this isn't fatal.
Whereas, and it's not that easy to catch. And in fact, for most of the countries of West Africa, it doesn't exist now.
But if you went to Africa, you got bitten by a black fly, it was blood there, you came home, a month later there was a nodule.
Oh, that's
Then you'd go take Ivermectin.
Yeah.
Would you have the nodule excised?
Sure. You bet.
Especially if it were in the middle of your forehead.
This is exactly right.
Which is, I understand how this was discovered.
Yeah.
This man, Robles, found nodules on the forehead of a small boy, and he dissected them and found the worms.
Bingo.
I guess that would be the thing to do, right?
I would think so.
Anything else we need to mention about onchocerciasis?
Well, it's on the wane.
Because of ivermectin?
Yes.
And vector control.
And because of the cooperativity of most West African countries, even during wartime, they would allow the airplanes that went in to spray the upper regions of the rivers for black flies. They would allow that to happen before going back and killing each other.
Sure.
So it's quite a remarkable infection. And this is one of the few parasites that has a real chance of being completely eliminated from at least Africa. But like I said, there's now a resistant strain of Onchocerca, so you have to worry about that. Better do it quicker.
So we want to eliminate it because it causes blindness
Yep.
in a good proportion of individuals, high numbers.
That's right.
And this, Dickson, is another one of the many parasitic burdens that
Correct.
we have.
That
we
is
need
exactly
to get
right.
rid of it.
I wanted to also mention the fact that this parasite was fostered and spread by dam building in West Africa.
Ah, yes. Let's talk a little bit about that.
So you have a huge, big, slow-moving river, and the next thing you know, you want to... Well, what were they using the electricity for, do you think, from these hydro dams?
Charge their laptop batteries.
That's a good guess, Vince, but that's not right.
The electricity from the dams to light homes, no?
No
Homes
way.
don't have
You're
electricity.
kidding? Of course not.
Military uses?
Keep guessing.
Commercial aviation?
I'll give you a hint.
Hospitals?
In Liberia, the country of Liberia,
I don't know. What would they
there
do with it?
was a company there where we used to send our medical students in third and fourth years to. And this compound was owned by a company. And the British would have pronounced it one way, and we pronounce it another way. It's the most common element in soil. There's a dead silence, folks.
I don't know.
What is the most common element?
What is the
soil?
most? That's a good Jeopardy question.
That's right, and I bet you Watson would have answered it too.
Molybdenum, selenium,
You're getting close.
titanium. No, selenium.
What is your microphone made out of, Vince?
Aluminium.
There you go.
I said it the British way.
Aluminium. That's right.
That's the most common element in soil?
It is. It is. And why is that true, Vince?
Don't know.
Yes, you do. You
The
just
comet hit us years
you're
ago?
not thinking. What is the valence of aluminum?
I don't know.
What?
Don't know.
Come on, it's 3. It's one of the few elements that has a valence of 3.
Yeah.
It's very, very, very tenacious. You cannot wash it out of the soil.
Okay.
So imagine living in a tropical zone.
You mean water can't wash it away?
That's right. So water washes all the others away, leaving aluminum. Where do you think aluminum comes from? They don't mine it.
Where is it?
It's all over.
So it's not mined, it's taken from soil, it's extracted? Because I know that Jamaica, they have
How
aluminium
do they
mines.
extract it?
I don't know.
Yeah, Jamaica too, sure. Any tropical country.
Reynolds Wrap.
That's right.
I don't know how they mine it.
Well, they scrape off the first 3 inches of dirt,
Yeah.
and they bring it into an electric smelter.
Okay, so that's where the power was going towards getting aluminum out of the soil.
And when they did that, of course, they created a new habitat below the dam for black flies to live.
Yeah, very cool.
So the aluminum workers were particularly susceptible to black fly transmitted onchocerciasis.
We do this a lot.
We do it a lot.
Full of stories.
We do it
All
a
right.
lot, Vince. And the dam, of course, creates a lake.
Sure.
And what do you think grows in the lake?
Black flies?
No. It's slow water. It's not fast-moving water. They need faster oxygenated water.
Other
Cold.
things grow in the lake.
How about malaria-bearing mosquitoes?
Sure.
How about snails that transmit schistosomes?
Which we'll talk about.
We will. So you've done 3 different things wrong.
Fast-running
And not only that, you've raped the environment of its elements through scraping off the top layers of this very infertile soil.
Yeah, which is the best part.
So you lose 4 different ways all at once, plus the workers all get sick. It's crazy stuff.
It says here, Dickson, that they've tried to make vaccines also.
They have,
and
they should make a vaccine against Wolbachia.
yeah, but there's some cases where Wolbachia is good in mosquitoes.
No, but I mean for people. It's not good in people.
No, but in certain, like in mosquitoes.
If they vaccinated people against Wolbachia and then you acquired Onchocerca, maybe you would cure the Wolbachia infection.
Are we sure that we don't need Wolbachia? Are we absolutely sure?
Well, we're never sure of anything, Vince.
Is it in our microbiome?
Death and taxes.
I don't know the answer.
No,
to that.
no, it's not. It's not. It's not. I can guarantee you it's
Okay.
not. I've looked.
Well, they. What was I going to say?
I don't know. We're easily distracted.
No. Wolbachia. Oh, you want to make a Wolbachia vaccine? I was just quoting your book here. They say that these vaccines have shown some promise, but you don't need it if this ivermectin is working, right?
I just need to tell the audience listening here, it's daunting to see someone who's not quite familiar with the subject sitting there reading your own book, and you get stumped on questions coming from your own book. That's not fair, Vince.
You should read it every night. I tell you, I do the same thing with my book. People ask me questions. I go, really? I wrote that?
I can't believe I wrote that. No, I wrote this.
Often my students in virology will ask me to explain some part of the book. And
Peter Hotez wrote most of the chapters on nematodes.
We shall have Peter Hotez.
We have to.
Well, we keep talking about it, so we're not going to talk about it anymore.
No, we're not.
We're going to just do it. I would like to read a few emails.
Let's do that.
We have always some good ones. The first one is from Betsy. Doctors Racaniello and Despommier. Hello.
Hello.
To start off, like so many of the messages you receive, thank you so much for this wonderful podcast. I'm a huge fan of both TWiP and TWiV, and have listened to every episode to date in both. It's fantastic how you present all of the information in a conversational way that is understandable for such a diverse audience of listeners. You two, along with your cohorts on TWiV, continue to feed my interest and very, very limited knowledge in the fields of parasitology and virology. I am a medical and biological illustrator/animator who became very interested in the field of parasitology while working on my graduate thesis at the Johns Hopkins University School of Medicine.
One of the best schools for illustration, medical illustration in the world. This was the first school for medical illustration in the United States.
My preceptor, Dr. Roel R. Dinglasan, an associate professor at the Johns Hopkins School of Public Health, who, besides being incredibly intelligent and enthusiastic, sparked my fascination with the malaria parasites and the field in general. And thanks to you, I can continue to stay up to date and expand my knowledge in this field. That being said, I thought you might enjoy the new issue of Johns Hopkins School of Public Health magazine. It's all about malaria.
Wow, great.
I really enjoyed articles and reading about what research is being done and also why some of the researchers focus on various aspects of combating this disease, such as control versus eradication.
Yeah.
Plus, there is the main illustration that beautifully depicts the life cycle of the malarial parasite that was created by an incredible medical illustrator, Jennifer E. Fairman. There is an interactive version of this illustration on the magazine site, too. To view the issue, please follow the link below, and we'll put that in the show notes.
You bet.
I thought you would enjoy this illustration, too, as you have mentioned aspects of my field on both your podcasts in the past. Specifically, you have commented on molecular animations by David Belinsky of Ex Vivo, who is at the top of the field in this area. You might already know about this, but in case you do not, the website, molecularmovies.com, showcases some of the most beautiful molecular animations out there. This site has a 2-part animation by another amazing medical animator, Drew Berry, on the life cycle of the malarial parasite. Don't despair, Dr. Racaniello. There are many animations on there about viruses, too.
Excellent.
Again, thank you for putting all of the time and effort that it must take to prepare and deliver such a fantastic podcast. I'm looking forward to all of your future episodes. Betsy,
great,
who is from weissbrodstudios.com. And P.S., in appreciation for your sharing of knowledge and enthusiasm for science, I've attached an illustration of mine for you of the malaria life cycle within the mosquito.
Cool.
I know it's not enough to repay you for your generous service, but it's a start.
Oh, nice.
Would you like to see it, Dickson?
Of course. Oh, lovely.
Plasmodium life cycle in the mosquito.
Oh, that's nice. That's lovely. That's great. One of your cohorts, Betsy, John Carapello, drew every life cycle in our book and won an award, actually.
From Johns Hopkins?
No, he got his degree at Ohio State.
It's a beautiful illustration. There, she signed it here.
That's very, very nice. And one of my best friends out there, of course, is a medical illustrator of some note.
Who's that?
Robert Demarest.
Oh, yes.
Who was the president of the Medical Illustrators Guild at one point.
Thank you, Betsy.
I love illustrations that tell the truth.
The next one is from Helen, who writes, I'm a med tech and a member of a group of people with immune diseases who is interested in helminthic therapy.
Right.
We listened with great interest to the TWiP on Trichiurus tricura. Trichiurus tricura, which some of us are already using. I wanted to ask you when you will be covering hookworm, and if you could discuss the therapeutic benefits of Necator americanus, which some of us are also using. I like the Trichiurus podcast so much, I'm going back and listening to all the old episodes.
Great.
I'm interested in how Triconella could be used therapeutically. I believe you said that if the larvae are injected directly into skeletal muscle, they would immediately infect the muscle cells.
This is true.
Would this mean that by bypassing the migration phase, the infection would not cause pathology?
Well, local pathology actually subverts the muscle cell into doing its bidding. So then I would consider that pathology, but it would be symptomless.
Mm-hmm. All right.
But that might not also be useful for people suffering from Crohn's disease and irritable bowel syndrome, because the immune system that works against the gut phase of the infection is not the same that works against the muscle phase of the infection. It's different. Mm-hmm. It involves IgE and eosinophils.
All right.
So Triconella hasn't been tried, but I know what you're driving at, because it's a long-lived parasite that would stay around longer than hookworm or Trichiurus, and therefore it represents a longer-term cure if it were.
Right.
But I don't know the answer to your question, no.
We did talk about this in the context of hookworm, right?
We did.
We talked about therapy.
We did, but I would like Peter Hotez to really address that issue, because he's a world expert.
All right, we shall.
We keep talking about we're going to have guests.
Well, we will when we get through the first descriptive phase, right?
That's right.
I have a family member with eosinophilic esophagitis, which is a disease that fits the hygiene hypothesis perfectly. Sudden recent increase in incidence, atopic disease, more common in children, and the eosinophilic response is supposed to fight off parasites. This is a horrible disease that causes much suffering and often leaves the person unable to tolerate any food, needing a feeding tube and elemental formula. Do you know anything about the incidence of this disease compared to the incidence of parasites worldwide?
I don't. I'm sorry, I don't. In fact, I haven't heard of eosinophilic esophagitis ever. I'm sorry that I'm ignorant in that field, but I can't know everything, right?
That's the end of the podcast, Dickson. You don't know something.
We're over. We're history. Vince, I could have started this podcast that way.
I'm just kidding. I'm just doing an elemental search in PubMed. There are just 2 articles with that term and parasite. Primary eosinophilic disorders of the gastrointestinal tract, a review from Gut in 2009.
They used to call those things tropical sprue and things like that. Non-tropical and tropical sprue, but I don't know what the current terminology is for those GI disorders.
All right. Thank you, Helen. The next one is from someone called Rich. Do you know a Rich?
I know several.
This one is Rich Condit.
I know Rich.
Rich says, "Hi, TWiP fellas. Hi, TWiP fellas. I should be working, but I'm procrastinating by writing you. I'm finally hooked on TWiP."
Good man.
Oh, that's a little play on words. Hooked on TWiP.
That's right.
Great show. Sorry it took a while. Until now, my taste in podcasts has been a bit narcissistic. I know this is a family show, but I really need to know if tapeworms have sex. You keep talking about eggs, but my sense is that these are not haploid, nor are they zygotes, but rather multicellular larvae encased in a shell, right?
No, Rich, I can aid you in your further understanding of tapeworm biology by telling you that they do have sex, but with themselves. A tapeworm is a large colony of single organisms connected together in segments. Each segment represents a unique organism. Okay? The head of this parasite is called the scolex, and then there's this little germinative neck region which germinates the segments. And each segment can break off at the end, and it contains the eggs, and it matures along the way. So how does a tapeworm have sex? And it does so by folding back on itself. Mm-hmm. And one segment will then exchange sperm with the other segment that it lies adjacent to, and they will transfer sperm across through their genital pore. Mm. And that's how sex is created. And you're right, they're diploid cells, and they're not larvae to begin with. They're single cells that are starting their embryonation process.
Okay.
So that by the time the segment matures at the end into a gravid proglottid, as it's called,
right,
all of the eggs inside are now infectious.
Right. Okay. Then he goes on. So, do tapeworms have a haploid phase? Are there sexes? No, they're just multisexed.
That's right.
Assuming there is a haploid phase, when, where, and how during the life cycle does fertilization take place? When do they self-fertilize?
When the segments go from an immature stage to a mature stage.
So the long adult worms do that, right?
It's about halfway down. If it's a worm 10 foot long, which tapeworms can achieve even longer than that, I would say it's about the 5 foot mark. That's where the segments are busy exchanging.
Yeah. Well, once it's long, it's difficult to do that any longer, right?
Oh, it's all wrapped up inside your gut tract.
It is wrapped. It's not just one line.
It's not just one. No, gosh, no.
If tapeworms are hermaphrodites, do they self-fertilize, or is there a chance to mix it up with other worms or both?
Well,
do they mix it up with other worms?
I guess they could if there were more than one worm there, but it's odd. It's an odd finding to find more than one tapeworm per person. So whoever gets there first probably commandeers most of the resources and prevents the next from establishing. Okay. Although I did see a woman once as a technician here. I was part of this team, and I think I described that, where we saw, I believe it was 9 different tapeworms in the same patient.
Yeah, I remember that. We have a picture of it.
And they were weird tapeworms because they had characteristics both of the beef tapeworm and of the pork tapeworm.
Yeah. All right, that's from Rich, who is our co-host
Thanks, Rich.
on TWiP.
Remember, parasites are just bigger viruses. Yeah. That's one way to look at it.
Next one is from Laura. Hi, I'm Laura from the UK, studying zoology and wanting to continue onto a Master of Sciences in parasitology.
Great.
I was wondering, are there parasites of parasites? Would Microsporidia be a candidate for this?
Microsporidia is a parasite of another parasite. Only Earl Widener would know for sure.
But there are parasites of parasites.
Oh, sure. Right? Remember, we've got double-stranded RNA viruses in Leishmania, and we've got some in Giardia as I recall, and we've got some in blocking on the other organism that viruses have been found in parasites. But
we've talked about this either
Yeah.
last time or the time
we have.
before.
It's not a common finding, but and it apparently, at least in terms of the visceral, or rather cutaneous leishmaniasis, it seems to favor the pathology of the infection. When you remove the virus, it ceases to become as pathological.
Now, microsporidia are now known to be fungi. They're
Right.
parasites, and they can go into bees, and together with a virus, remember that they think this might be one cause of colony collapse.
Yeah.
So,
And we know they're fungi because
that doesn't answer her question though.
No, it
because
doesn't.
She wants to know.
I know.
Is this is a parasite, but does it parasitize another
parasite,
ever?
right.
We don't know the answer to that.
I don't know any examples of it.
Yeah.
Let's put it this way. But I only pay attention mostly to the ones that infect humans, and there's an enormous spectrum of parasitism out there waiting to be discovered. Virtually every non-parasite has a parasite.
Okay, next one is from Ryan. Greetings. Just wanted to drop you guys a line and let you know I enjoy the podcast. I found TWiP while actually looking for a podcast about photography.
I love it.
But gave it
At
a
least he
listen.
wasn't looking for pornography.
He gave it a listen anyways. I'll be downloading every episode from now on. Parasitology was my favorite class as an undergrad.
Where did he go
Thanks
to school?
for the work you do. I don't know. Tell us, Ryan.
Yeah, Ryan, write back and tell us where you went to school. I'd like to acknowledge your teacher.
This week in photography, this week in parasitism.
This week in pornography, this week in...
What I like is that he said, ooh, cool, let me check this out, and he
Right.
liked it. Instead of saying, no, this is not what I'm looking
No, you're
for.
quite right.
He had an open mind.
This week in politics.
Could be. Thank you, Ryan. I think that's a great story. And the next one is from Todd, and we'll end with this one. Hey, docs, I'm a computer programmer who listens to podcasts on my long commute. The highest science education that I've had was college chemistry. As an electrical engineer, the science classes we took tended to be not biologically oriented, so while some of it is over my head, to say that I find biotech and molecular biology fascinating is an understatement.
Wow.
My question.
Nice.
I was born in 1970 in southern Louisiana.
So was I.
Not in 1970.
1940.
My uncle had a horse, and me, my dad, my uncle, would scoop the manure out of the stall and put it into a pile on the side of the horse barn. Some months later, after the manure had been exposed to the weather and sun-dried, we'd scoop it into the wheelbarrow and put it along with composted grass clippings in the garden as we tilled the soil for planting. I was wondering what kind of worms and or other parasites we might have been exposed to by using horse manure to fertilize our vegetable garden. If I'm understanding things correctly, I don't think there is any danger for worms lasting long enough to get into our veggies and infect us. But what about direct contact when handling the sun-dried manure? Shovels and gloves, but still stuff happens. And is there really anything to fear other than worms? I am in good health, so I think the answer is probably no. I'm just learning for learning's sake.
Well, I have an answer for you. The answer is that there are no parasites that you can catch from horses in their feces.
But there are parasites.
Yeah, one of them is called Despommier. It's their version of pinworm.
Oh. It doesn't go to people.
It doesn't go to people, and people's pinworms don't go to horses. In fact, cow parasites don't go to horses.
Or people.
And horse parasites don't go to
So that's
cows.
why we can use cow and horse manure to fertilize.
Safely.
But he's worried even when
Yeah.
it was fresh, that's not a problem,
No.
right? So you're okay, Todd.
And so are all the horsemen.
Mm-hmm.
You know that little ditty, I presume.
No, what is it? Tell us.
She was only a cattleman's daughter, but all the horse manure.
Dickson. I call you Devin, my son. Dickson. Okay. P.S. Lately I've been listening to Fib, TWiV, and TWiP podcasts to the point where I'm neglecting my techie podcasts. You guys are causing me to lose some of my geek cred.
Too bad. Join the other geeks who said we're not geeks.
Isn't it geek to listen to science podcasts?
I think it is.
Why not, Todd?
Come on.
Not tech, but cool. All right. I think we'll stop there. I'll make a note in the show notes so I remember which ones we've read and save the others for next time. There was just one more here I put in, but we'll save it for next time because now Dickson is going to be next to me and we can podcast more frequently.
That's right. I'm moving into your quarters, Vince.
Or maybe less frequently. I'm just kidding.
Maybe we get sick of each other and stop podcasting altogether.
If you like TWiP, tell everyone about it and subscribe in iTunes. It's free, so you can get every new episode as they are released. You can always download episodes at microbeworld.org/TWiP, where we keep show notes, and you can also play the episodes right there. If you do use iTunes, by the way, and you haven't already done so, put a review in. Just has to be a sentence or two saying you like the show. It helps us keep us on the front page of the medicine directory in iTunes. If you want to listen on your smartphone, either Android or an iDevice, you can get an app. An app, and you can get microbeworld.org. They have a link to that.
Maybe we should have a parasite app. Somebody, by the way, wrote me recently from Tennessee. I'll think of where in Tennessee. He wants to do a phone app for the identification of parasites.
I think that's a good idea. I always wanted to do one for viruses.
Yeah, I think it's a great idea.
But
he wants me to help him. I'm glad to do it.
I think you should, since you have plenty of time.
I have lots of time now. Thanks.
I'm just kidding. Send us your questions and comments to twip@twiv.tv. Dickson Despommier, thank you so much,
Vince,
as usual.
It is my pleasure.
Dixon is at triconella.org, verticalfarm.com, and medicalecology.org. I am Vincent Racaniello, of course. And where am I, Dickson?
You are at virology.com.
No, they're virology.ws.
Oh, okay, .ws.
The com was taken.
Too bad.
In 2004 when I went looking.
The nasty virology.com,
virology.net, virology.tv, they were all taken.
Gee.
So I got virology.ws, which is from Western Samoa. And there are many Libyan websites, .ly. A very famous one is bit.ly, bit.ly. It's a URL shortener. And if Libya goes under, which it might,
over. It's going to go over, Vince, not under. Gaddafi goes under.
We might lose all of these.
No,
websites.
It'll get better, not worse.
You have been listening to This Week in Parasitism. Thanks for joining us. We will be back soon. Another TWiP is parasitic.